Yes. Well, before we start, let me first explain why we have this meeting today. The story begins with like a few months ago, I read Jason's paper and ran his code. I find his paper is really interesting. The code is really reproducible. I found a few problems and I reported it to Jason. He nicely answers my question, and yeah, we have a small talk about this work, and I asked him. I find this paper is really interesting, but I find a lot of the mathematical details is really confusing. So I asked if he wanted to give a talk to explain some of the mathematical details, especially for those people who have no such a strong mathematical backgrounds like us, like me. So like 2 months later, we have this meeting. It's really honored to have all of you guys join us, and also have Jason to talk about his paper. Before we start, let me first briefly introduce Jason. So Jason is a second-year PhD student at MIT. Uh, Previously, he was a research engineer at DeepMind, and he obtained his bachelor degree from Johns Hopkins University in computer science and applied mathematics. I guess a lot of the, I guess a lot of the works like AlphaFold multimer and RF diffusion and this talk, the Framediff, are all from Jason. So Jason is really a talented, excellent, exceptional researchers in this field. So we are really honored to have you today. And without further ado, let's get started.
Thanks, Shang-Chi. You got to let me share the screen. I'm unable to do so. Okay, can everyone see?
Yes.
Okay, great. Yeah, thanks so much for the introduction. Glad to be here, and thanks for the interest in Framediff. So hopefully I can dispel some mysteries about it and at least give some intuition about what's happening in the method and stuff. So actually the title of my talk is Diffusion Models for De Novo Protein Design. Because I mean, just broadly, we've been working a lot on diffusion models for this de novo design problem. Although Framediff is just for protein structure generation. So let me see. Okay, so just to motivate the problem a little more, nature has produced lots of proteins that support life, but as humans, we create a lot of challenges that evolution can't work fast enough to solve. That's why we're interested in de novo protein design to directly engineer new proteins for a lot of the challenges in the world. Some of these challenges, they fall into making new vaccines or creating better enzymes to degrade plastic, or biosensors for like detecting viruses, and also maybe like most popular is genome editing that you can do to modify DNA. So when we think about protein design and how machine learning plays a part in it, there's like these 4 steps that I and a lot of other people think about. So the first one is called backbone generation. This is where you're trying to generate the 3D structure and what our work is about. And then there's sequence generation, which given the backbone, you're trying to generate the sequence that can fold into it. And then there's virtual screening, where after you generate the backbone and the sequence, you can use AlphaFold or some other method to pick out the best designs that you came up with and test them out in the lab. The last one is that once you have maybe like a good sequence and structure, you might want to optimize it for a certain property that you want to have in the protein. So for each of these, there have been a lot of research in them, in particular using deep learning. The backbone generation, previously before diffusion models, there wasn't a very good method to do it. So one of the most exciting things about diffusion models is that we can achieve really amazing results in backbone generation. And And then ProteinPNN, if you've heard of this work, works very well for sequence generation. And then, all the new protein folding methods, AlphaFold or ESMFold, they've been state of the art in virtual screening. And then lastly, directed evolution is a Nobel Prize winning idea for property optimization, but people have been thinking about new machine learning methods to solve this problem too. So just we're going to today focus on backbone generation, but it's good to keep in mind the full picture of how one would do protein design from end to end. So these are the four problems I'm going to solve, but what I'm going to argue for is kind of a probabilistic viewpoint, a probabilistic way to frame the problem and tackle it. So for instance, for the first problem, we want to generate a distribution over backbones. So that's what the probability of BB backbone is, but we also might want to do conditional generation where you're trying to generate the backbone conditional on some property that you want out of the structure. And this property can be many things, such as binding to another protein or having higher stability or stuff like that. So once you have the backbone, you definitely need to get a sequence for the backbone. So then ProteinPNN is a way to sample the sequence conditioned on the backbone. And once you have both of these, you can feed that into AlphaFold 2 to get the designability score. And you can also measure fitness as a probability distribution as well. So the focus of this talk will be just the first step of how to do general models over the backbone. But just a little bit more motivation is that we want to do probabilistic modeling because first we want to do exploration so that we can generate multiple designs. Second, the probabilistic way allows us to use like Bayes' rule to combine and reuse methods. And this has also been one of the most exciting things about diffusion models is that you can do really good conditional generation with them. You can guide diffusion models and they're very good at generalizing beyond the training set. And then probability, it's the language of stochasticity. So you can calibrate these models with the biological noise that's present in the data set. So this is also very useful too when you're doing design problems. So first, I'm going to talk about Framediff. And the crux of Framediff is how we developed SC3 diffusion, a new structured way to do diffusion over protein backbones. And then I'll just very, very briefly talk about RF diffusion because these two kind of go hand in hand. RF diffusion is basically we took SC3 diffusion and then we combined it with Rosetta Fold 2, a very good protein folding method. And then the main advantage here is that we were able to do conditional generation to generate structures conditioned on certain properties that we wanted. So yeah, I'll first go over Framediff. This was done with some amazing collaborators. A lot of different universities. So Brian was at Columbia University, Valentin is in Paris, Emile is at Cambridge University, Arno is at Oxford and DeepMind, and Regan and Tommy are my advisors. This was published at ICML 2023, which just happened last week. So I personally got excited about diffusion models in 2021 when these models started to come out, like DALL-E 2 and ImageGen, and they're really kind of mind-blowing to a lot of people because they had this aspect of creativity in them, in that they were able to generate things that look super realistic, but were able to kind of imagine new images and new sentences. So for instance, DALL-E 2 and ImageGen, what you do is that you give it a sentence and then the model would generate a different image, and it could generate multiple images too that would kind of follow what the text was trying to say. So this was really amazing at the time. And at that time too, I was thinking, why can't we do this for proteins? Because this is exactly what we want to do when we're trying to design novel proteins. So I actually didn't plan to go through the technical details of diffusion models because there are a ton of different tutorials out there. And I think time would better be spent talking about like more technical details about proteins. So I'll give a very brief idea of what happens in diffusion models. So you have a variable x. This x is interpolation between time, where at time 0, you have your data distribution, and then you iteratively add noise until it goes to the very end. So like at xt, there's going to be a certain amount of noise added, and here is just Gaussian noise. And then the goal is that at the very end, so capital T, you're going to end up with a complete noisy image. And the idea is that you can train a neural network to go backwards, where starting from the end, if you train a neural network properly, it's going to iteratively remove noise until it ends up back in a data distribution. So you can think of it as an iterative refinement process that the model is slowly removing noise until it gets to the true data point. Okay. So why do we care about protein structures? Structures are important because this is what determines the function of a protein. So here, for instance, is ATP. The structure determines how it works. It generates energy in the body, so it's one of the most important proteins that we know. And de novo protein design is we want to come up with new protein structures for new functions that I want. So there's an element of creativity here that we had to discover new structures. So if we want to do diffusion models on structures, the first question you have to ask is, what kind of representation do you want to use? Now, there's a lot of different options. The first option that might be the most obvious is that let's model every 3D atom. So here I have a protein backbone, and just a little bit of background of protein. Also, I'm going to assume that people kind of know what a protein is, I guess. But let me know if you don't. A protein is made up of a lot of residues, and every residue has the common structure of 4 atoms that comprise the backbone. And so you can see that it's copied over a lot of times, and you have the choice of modeling every single atom independently. So by doing that, you can move every atom, but when you do that, then the bonds are breaking a lot, and it's really hard to model. The other option is to only care about the torsion angles. So if you kind of look at the backbone, you'll see that there's this repeating pattern of 4 atoms and then some torsion angles between them. The issue with this is that although you have less degrees of freedom, you have this something called the lever arm effect, where if you just rotate a single bond a little bit, that might cause a huge shift in the actual structure and how the atoms are arranged. And so because of this, optimization cares a lot about having smooth surfaces to optimize over. And so trying to train these models on torsion angles just never really works out because it's too unstable to train. And then something that's in the middle that has become really popular ever since AlphaFold 2, which was to use something called the frames. And this is something in the middle between the 3D atoms and then the torsion angles, in that you're still maintaining a lot of the bonds, but you have control over the atom placement because every frame has a translation, but then it still has this rigidity that that lets it like not have too much freedom in how it breaks the bonds. So this might seem like a good solution to be in the middle of angles and positions. However, it comes up with a cost, which is it has a more complicated math structure that you have to deal with. And so this was what led to SE3 diffusion. To go a little bit more into how you construct this frame, let's kind of take a closer look at a single residue. So again, like this is what a residue would look like. It has 4 atoms. And then what we do is that we'll do an operation to convert this into a frame. So the C alpha position would be the center of the frame, and this would be the translation. So it's a vector in R3. And then you look at the vectors like V1 and V2 to the neighboring carbon and nitrogen atom. And then from this, you can do Gram-Schmidt to get this 3rd orthogonal vector. And then from that, you have 3 orthogonal vectors that comprise of a rotation matrix. And so an element of SC3 is just the translation vector of C alpha and in the rotation around C alpha. And so you see that we kind of ignore anything about the bond that connects C alpha to its neighboring atoms. And that's a win for us because then we've removed degrees of freedom by only caring about the rotations. It makes training a lot more stable. There's also this torsion angle here that you can either predict or just impute. It just controls where its oxygen placement is, but this is very stable, so it's not that difficult to predict. By the way, if there's any questions, just feel free to shout or put something in the comments. Yeah, happy to take questions along the way. So that's how we construct one frame. Because proteins have a lot of atoms, a lot of residues, you would have multiple frames along the chain because the protein has a train-like structure. And then because proteins are very big, you'll have lots of frames. And so here I'm visualizing the frames in Matplotlib. The line between them is the actual sequence, the training structure, and then along them are just the rotations and translations of the frames. So we got here, and so a frame is represented in SC3. So the technical question is, how do you do diffusion on frames? So this is important because it's not E3. If it was E3, then you'd have like chirality issues in that you wouldn't be aware of the reflections that would happen. But we want to care about the reflections, so that poses a challenge. And because of that, in mathematical terms, what SC3 is that it's a semigroup. And so that's what this symbol represents. It has this X and like a bar here, which means that it's a semigroup with the group action that looks like this. So what's important to note here is that if you compose two SC3, so two frames together, Rx and R prime X prime, you end up with this composition element here. And the important thing to note is that these two things are not independent, and that when you compose these, there's this rotation that gets applied to the second translation. And so this causes issues because when you have this structure that is not independent, there's no canonical diffusion that you can just grab out of like probability theory and apply to this method here. That's the main issue of why it's not straightforward to do diffusion on SC3. So our goal in the paper was to parameterize SC3 in such a way that the rotations and translations are independent and still kind of satisfies the properties of STEs. And the goal was to not go too far into theory, but construct something that was kind of simple that we could train a model over. So there's always this tension between theory and practice. Of theory, you want something that's nice and has an elegant structure in math, but you don't want it to be too theory-based. It still needs to be very practical to be able to train a model on neural network with. So the goal here was to have something that was satisfying the properties of diffusion models, but something that we could train on with train with the neural network very well. So in particular, what I just described previously was that if you look at it from a Lie group, then it has this weird group structure. And because of that, there's no like easy way to grab a diffusion and do diffusion over it. So that's called the Lie group perspective. I'm not going to go into details of that, but something you could look up later. So because there's no easy way to do diffusion, we have to manually construct this diffusion on SC3. So the first step to do this is that we want to define how do you do Brownian motion, or how does like noise propagate on SC3. So like, let's say here, M is SC3, and this is like a manifold. So it's like this circular looking thing just for visualization. But we're going to describe diffusion as happening on the tangent space. And the tangent space is just a plane that's defined by the tangent vector, which is where the gradient is pointing towards. So you create this plane at every point on M, and you take a step along a direction. And by doing that, you're going to be able to move along this manifold and do diffusion over it. And so that's what this TX of SC3 stands for, is that you want to do diffusion on the tangent space of this manifold instead of the manifold itself. Now, when you look at this manifold, you have a choice of what your distance is going to be. So in Euclidean space, distance is very simple. It's just, the L2 distance between 2 points. But here, you have to be more careful about how you define distances on manifolds. And so I'm not going to get into details of this again, but there's two choices. There's one choice of being left invariant, another choice of being right invariant. And the crucial thing is that in Riemannian geometry, for SC3, only the left invariant metric kind of factorizes into something called the product manifold. So if you're very curious about the math, you can go into the appendix and also this reference to learn why this is the case. But by only choosing the left invariant metric, you can decompose SC3 into this product manifold of SO3 times R3. And so the main thing to note here is that this doesn't have this bar on the side, which means that when we do this composition inside of the metric, they decompose. So let's just like slow down here. Here, when we do this composition inside of the metric, it decomposes into something that's very independent, that the rotations are by themselves and the translations are by themselves. Previously, if you go back to here, if we just decompose them not in the metric space, you can see that there's this relationship in the x's that there's this r here. That's just really annoying, and it's hard to do diffusion over. So it is this idea that in the metric that if you use this left invariant metric, that there's this independence property that you can now do diffusion separately on SO3 and on R3, which is the nice property that we were looking for. And this independence also leads to some further results of trying to show invariance of these measures. That's important for proving equivariance of the SC3 diffusion. So yeah, hopefully that somewhat made sense. Okay, so we kind of achieved our goal, is that as a Riemannian manifold in the metric space, that it's this product of two manifolds that we can do diffusion over. And because of that, we can do diffusion independently, that we can diffuse the translations and we can diffuse the rotations separately. And so let me just play this again, that if I take the ground truth frames and if I apply diffusion on them, then this is kind of what the process looks like. You can see that the train structure starts to break and the frames are rotating, and then it eventually starts becoming crazy until you can't really tell what the original frame was anymore. So this was the goal that we set out to achieve. To just like go a little deeper into each of the diffusion components, the translations kind of look like this. It's just normal Brownian motion, that's how normal diffusion models work. In particular, we do the VP SDE, which is the variance preserving SDE. And so it kind of looks like this, where you would have like your data, and then as you just keep noising it, it ends up looking something like a standard Gaussian at the end. For SO3, we have to utilize a different distribution called the IGSO3. So IGSO3 is like the analogous normal distribution on the SO3 manifold. So some Russian guy figured this out in a paper back in like 1900s that we pulled out of, but it's still useful today. And so what I'm going to go into in the next slide is to explain a little bit about SO3. But here, what you need to know is that IGSO3 defines how diffusion works in SO3. At time 0, you would have the data distribution, which is a perfect rotation. And then as you go to higher time steps and you start to sample from this distribution, it's going to become noisier and noisier until it's a completely uniform distribution on the SO3 manifold. So some background on SO3 of how to like interpret this manifold. There's three objects that you need to be aware of if you ever work with rotations. The first is the rotation matrix. The second is the rotation vector, and the third is quaternions. And so all three of these are related to each other by a series of operations. In that the manifold is a rotation matrix, which we know, and then you apply the logarithmic map to get to the tangent space. And the tangent space, which is a Euclidean space in d dimensions, is a rotation vector for SO3. And then for rotation vector, you can go back and forth between the skew symmetric and the quaternion. And so traditionally for Riemannian manifolds, if you want to do this mapping between the manifold and a tangent space, it's quite expensive. It's actually an infinite sum in the power series. But for SO3, it's special in that something called the Rodriguez formula gives a closed form mapping to go between all three objects here. So it's a really elegant manifold that is really easy to use and really easy to code up as well. And all this math is in the Framediff code base as well. So yeah, and each of these three objects has a use case in Framediff. So the first is the rotation matrix. This is useful because if you want to compose two rotations, you just multiply them together with matrix multiplication. But the disadvantage is that we can't add them together. If you add two rotation matrices, there's no guarantee that they're going to still be orthogonal. So yeah, you can't do that. But we need to add rotations together in order to do Brownian motion on. And so that's where the rotation vector comes into play. So this is the vector in R3, and this allows for addition that you can do Brownian motion on. The issue with that is that it's degenerate, and I'll go into details about that later. The third object is a quaternion. So quaternions is very related to a rotation vector, but it has a homogeneous coordinate. So that's why it lives in R4 instead of R3. And the benefit of quaternions is that it's just known to be numerically stable. people have been done some experiments of like what is the best representation on a neural network for rotations, and quaternions, it turns out to be really stable for learning. So that's why if you look into the code, if you also look in AlphaFold 2's code, they actually predict and learn quaternions instead of rotation vectors or rotation matrices. And so our model is going to predict quaternions, but we can map that to rotation vectors. And then rotation vectors we can do diffusion on. But then if you ever want to like do multiplication, then we'll go to the rotation matrices. So what you'll notice is that just like in R3, where we do addition, multiplication, subtraction, we can do all the same things in SO3 as well, as long as you can go between all three of these representations very easily. So to get a little bit of intuition about Brownian motion on SO3, I mentioned that IGSO3 works on the tangent space, so the rotation vectors. And so rotation vectors, I'm coloring in red, and then the rotation matrices, I'm coloring in blue. Like before here, to go between these, you need the logarithmic map. And here again, I'm just showing that, yeah, there's this relationship between the two in the logarithmic map. So a rotation vector, you can think of it as having two components. So if R is the vector, it has this omega, which is the magnitude of how much you spin, and then this E, which is the axis that you spin around. So you can represent any rotation in this way of having an axis and a magnitude of how much you spin around that axis. And so again, Rodriguez formula gives us a way to go between these two objects seamlessly. So IGSO3, it operates on the rotation vector space. And you can think of it as the heat kernel on SO3, and it has this form, which is an infinite sum. But in practice, we just truncate it to only have about like 1,000 terms or something, and it still is a very good approximation. And again, just showing that if you look at this distribution, and depending on what your T, which acts as the variance in this case, higher the T, the more kind of like wild the rotation vector gets, so it becomes more uniform as you get to higher T. Okay, so that concludes the part about SO3 and SE3. So we do the forward diffusion, and then we train a neural network to learn the score of this distribution, which the score is just a lot of like algebra and calculus that I'm not going to spend too much time in. But if you train a neural network to do this, then you can train a neural network to go backwards, starting from pure noise in the rotations and translations, all the way back to time zero, which here you can see that all of the frames are nicely lined up. And we can do a canonical mapping to get to the all-atom representation of the backbone here. Okay, ooh, I messed up here. Okay, so the other thing that's important is invariance. In that, like, if I start with noise and if I rotate it, then if I do the sampling, then it should lead to the same protein at the end. And you want to make sure, so there's like some theory about this, is that you need to have an invariant density and an equivariant score. So this is just spelled out here. And then in the forward process, we want the rotation to be invariant. And this is just given to us by the density that we use, the IGSO3 density. And then for the translation, what you have to do is zero centering. So this is something that was figured out by GeoDiff and also equivariant flows. But we just kind of recapitulate here for SE3 that you have to do this zero centering to have translational invariance. And then if you have both these to be invariant, then the SE3 part is also going to be invariant. Now for the reverse process, you need an equivariant score for the distribution to be invariant. And so that's another theoretical result that's in our paper and also in previous papers in GeoDiff. And so what this practically means is that when you go backwards, you're going to have this projection that will always zero center the translations. And then as long as the equivariant score is learned properly, then you're going to have an equivariant distribution that you learn. So the neural network that we use here is called Framediff. So the architecture is kind of laid out here. I'm not going to go into details, but the main thing is that we use IPA, which is an invariant point attention network. That is SE. So yeah, that should be SE3 equivariant. And this is also used in AlphaFold 2 as their SE3 equivariant structure module. So, but because here we're not using MSAs or sequences, I decided to instead put a transformer inside of this neural network so that the model has a way to do geometric reasoning in the attention, but also to do sequence-based attention as well with the transformer. And so this I found to be quite important to have this transformer component in here for like stable training and for high performance. Another detail of this model is that IPA was traditionally designed to predict the clean data. So it's designed to predict T naught instead of the score exactly. So we leverage that fact to predict the true frame, but then we can calculate the score by using this predicted frame instead. So instead of other diffusion models that predict the score directly, we instead predict this clean data and use the clean data to go to the score. And then the score is the loss that we use here in the denoising score matching loss. Yeah, so that's the model. So for evaluation, a lot of people always have questions about this, like how do you evaluate generative models? So the quick and dirty answer is that there's no great way to evaluate generative models other than put them into the lab and test them out, basically. But because that's expensive and we don't want to waste too much time and money, we need to do an in silico method, which is something that's done on the computer. So what we choose to do is something called the designability test, which is where here we'll first use Framediff to sample some backbones. And then because the backbone doesn't have a sequence, you have to design the sequence using protein PNN. So we design a bunch of sequences, which is this variable here. So in the paper, we use 8. We design 8 sequences. We fold them all with ESM fold. And ESM fold is going to give a prediction of what the sequence it thinks it is. Sorry, the structure of what it thinks the sequence comes from. And then we can compare the final structure of ESM fold with the structure that comes out of Framediff. And then this we call the SC RMSD or the SCTM, depending on whether you choose RMSD or TM score. And different people have different opinions on which one is better. SC is just kind of historical reasons where we used to call this the self-consistency test because, yeah, you could think of it as like a consistency metric between ESM fold and Framediff. Okay, yeah, so you can think of this as like testing the realism of a protein backbone based off what ESM fold. And you can also use AlphaFold here as well. It's just way more expensive, and ESM fold is just convenient to use. So here are the results, the quantitative results. We sampled a bunch of proteins at different lengths, and then we calculated this designability metric. You can see that for really low lengths, like 100 to like about 300, most of the time the structures are very designable. And so, yeah, we use 2 as a cutoff. So if the SCR-MSD is less than 2, which means that the structures are almost perfect, then the model gets high success rate. However, as you go up and higher to like the 350 to 500 residue range, the designability score starts to go down. Yeah, and you get a lot worse performance. And we also color them by the SCTM. So you can see that the SC-RMSD test is way more rigorous than this SCTM test that some people do. We also analyze the secondary structure to show that the model is able to generate like a wide variety of secondary structure elements. So there still is like a bias towards helices, just because in PDB, which we trained on, there's a lot of most things are helices. But you can see that a lot of things are also mixed. There's mixed helices and sheets, and there are some samples that are just pure helices as well. Yeah, I'll just note that something that we noticed is that for the longer proteins, especially, the model has this tendency and bias to sample only helices. And so I'm not sure why, but it might be that in the data set that a lot of these large proteins have mostly helices in there. Okay, so for yeah, some more results. So another thing you care about is that, okay, you can tell me that it's designable, but is it useful? As in, is it actually generalizing beyond the training set? And so here we show that that is the case, at least for some of the samples. So this is the designability metric that we use. And so you want things that are to the left of this 2 bar. And this is the PDB TM score, which is the TM score to the closest example in PDB. And we calculate this using fold-seq. And so like 0.5 is the cutoff of like determining if 2 folds are very similar to each other. So we see that there are some examples in this lower left-hand region on proteins that are both very designable, but also very different than things that are in the PDB. So we're visualizing 2 of them here. the thing in red box and the thing in green box. The thing in the red box is kind of a short protein. It's length 135. And ESM fold has a very high confidence of it. So it thinks it's a realistic fold that could happen in nature. But then if we look into the PDB and find the closest matching structure, we find this thing, which is very, very different from it. So it's evidence that Framediff is able to kind of extrapolate beyond the training set and generate stuff. And we show that it also happens to the longer proteins as well. So here there's a 460 length protein. Like again, it's all helices. But interestingly, the way that the turns and loops happen is very different than anything in PDB. So this is the closest thing that we found, and it has a PDB TM score of 0.5, which means that it's very different than things that are in PDB. Okay, so here are just like animations of like what the trajectory looks like of how the protein is slowly generated across time. So these are like small proteins. We can see that there's still like a wide range of structures that come about. And a really interesting thing about it is that you can like visualize how the model kind of predicts like the very coarse structure, and it starts doing like fine-grained details. So in particular, for like instance here, like the secondary, the data sheet doesn't like really get the spacing right until the very end. I guess it's still trying to figure out like which ordering they want it to be. So it's kind of like an interesting way to look at what the model is thinking as it's generating the protein. Here it is for like longer proteins. So these really big proteins, there's way more complexity and variety in how it's being generated here. Yeah, and I'll just give a shout out to Arian, who made this awesome tool to visualize these and make these visualizations in VS Code. Another thing that we noticed that was quite interesting was that the noise scale had a very big important impact. So you can generate, so when you do reverse diffusion, it depends how much noise you're injecting into the model at each step. And so this has some like theoretical background for like high robustness and stuff. I think someone, Mike. Okay. Yeah, so noise scale, it can kind of ruin or break your samples. So we found that if we use a noise scale of 0.1, which means like less noise is injected, then we can get very high quality samples out that are more designable. So yeah, across like the different lengths, the samples look very like they have a lot of secondary structure and not many loops. But then if we use a noise scale of 0.1, then there's less secondary structure and more loop, and it's like more flexible components in there. So it's like this interesting trade-off of how high quality do you want to be, but perhaps less diversity, or like do a lot more exploration and generate more novel folds using noise scale of 1.0. So conclusion, when we started to do Framediff, what we wanted to do was like take a lot of ideas from AlphaFold 2 and then turn that into a generative model. And so diffusion was one way to kind of take the elements and make a generative model. So yeah, we follow kind of the best practices. One of the defining features is that we use frames that works really well in AlphaFold 2, and also its structure network, which is IPA, as the equivariant score model. So some directions and limitations is that eventually want to, of course, be able to generate the sequence and the side chains all at once instead of doing a multi-step process. And then second thing is like conditional generation, such as motif scaffolding that I actually did in like a very first paper on diffusion models. So yeah, on this last point though, the conditional generation, like before we started to get working on that, we had a collaboration going with David Baker's group. So that's kind of what led to our diffusion, and that we wanted to do conditional generation actually for real protein design tasks. Yeah, this is a paper with RF diffusion. There's a lot of co-authors here, co-first authors. I think the paper actually has a different name now, so this is not up to date. However, like this just got published in Nature 1 or 2 weeks ago. Yeah, 2 weeks ago. So I'm not going to go into too depth because we've already had a lot of talks on RF diffusion. But I'll just briefly kind of mention like what the connection is between Framediff and RF diffusion, which is that when we made Framediff, we developed SE3 diffusion, and then in RF diffusion, what we wanted to do was combine SE3 diffusion with like a pre-trained protein folding model to do fine tuning on. And so it was kind of like this happy marriage between SE3 diffusion and a pre-trained protein folding model from David Baker's group, so Rosetta Fold 2. And so what the main difference with our diffusion was that it's able to do conditional generation. So we fine-tuned it to do one binder generation. So like given an epitope to generate a de novo binder for that. Another thing is scaffolding. So I given a motif to generate a scaffold around it. And then another thing was to do like different combinations of conditions. So one, to be symmetric, two, to be a binder, and three, to do scaffolding. So to do all three things at once. And this is something that's really interesting and powerful that you can do with diffusion models. So I'll just like kind of quickly say like how is it done, and the way to do it is being called diffusion guidance. And so this is also something that's given to you because we're doing the probabilistic method in doing diffusion, in doing generation. So typically how this would work is that you'd have your diffusion model, which is in green here. Like this is the score that you would use in the reverse sampling process. And you tack on this this guidance term, which in previous works, people have done it with like a classifier. So it's be called classifier guidance. But in this paper, what we do is we use the potential. So that's why there's a capital P here. And the potential, you can imagine, is like this force field around the molecule or the protein that kind of is like a higher region and a low value region. So we'll have like a very a low value region here if like if you want the protein to be very like not clash with it. Yeah. So if the protein ends up overlapping in this red region, they can give it a high penalty. Whereas if it's kind of farther out, then it's going to give it a reward. So it'll be blue here to give it higher probability. And so the guidance is a way to kind of guide the generation of the guided generation towards specific properties that you want. And so when we're trying to generate proteins that will bind to small ligands, you can see that if you don't use a guidance, then there's going to be clashes that it's not going to be in the pocket. But then if you use the potential, then it's going to satisfy those properties and like basically generate good proteins for you. So the last thing with our diffusion is that it's different in that we actually do a wet lab validation here and show that the proteins can actually be expressed and that the structures actually match what was designed using RF diffusion. So it's like electron microscopy. This is some very early cryo-EM renderings of like what the generated binders were, and then we can even see that the designs and the actual protein structure actually closely match each other in like subatomic accuracy. So yeah, acknowledgements. Framediff, I already mentioned that, and RF diffusion. A lot of people from David Baker's group, and also my advisors, Tammy and Regina from MIT. And these are just like some references. So like this is a very good introduction to Lee theory, and also a very good introduction to SE3. And then if you want to learn more about diffusion models, check out Lilian Wang's blog post for diffusion models. So yeah, that's all I had, and happy to take any questions.
Thanks so much, Jason. Excellent talk. First quick question was, what do you see as the difference primarily in terms of inductive bias between SE3 diffusion and RF diffusion, particularly because if I remember right, in your paper, you touch on like kind of the influence of pre-training a protein structure prediction model versus training from scratch. Like what kind of effects do you see like pre-training happening and like downstream effects versus like training from scratch?
Yeah. So when we're doing the pre-training, we thought that we would see like not much novelty in the structure that came out of RF diffusion. We thought it would just spit out things that were in PDB. So the thinking was that Framediff would have an inductive bias to generate things that were like physically plausible, but like a little bit more esoteric, like very different from things that were in PDB. But in fact, like RF diffusion kind of in a weird way extrapolated beyond PDB as well. So that was kind of an interesting finding with the pre-training that we did for RF diffusion. So yeah, surprisingly, they both share a lot of the same inductive biases, as in they both generate things that look like PDB, but then often they also generate things that don't like PDB. But Framediff, I guess, is interesting in that it doesn't need the pre-training to be able to do this, and that it doesn't need this evolutionary pre-trained to do it. Whereas RF diffusion, in our test, we kind of tried to train it from scratch, but it was never able to learn the diffusion process without this pre-training step. And I think that's just because it was a really big model that you couldn't train really well without the MSAs. So I guess how I view it is that Framediff and RF diffusion, they are very similar methods, except that one has a reliance on MSAs and has a stronger inductive bias from the MSAs that helps it to stay in the realm of proteins, but also remarkably still generalizes beyond PDB still.
Thanks.
Hey Jason, I have a question that have you tried that not diffuse on rotation, but just directly predict it as the final step? And is this has big difference between just direct predict rotation and diffuse rotation?
Yeah. Yeah, it's an interesting comment. So I think the closest work to that is Genie by Al-Qarashi's group. They kind of don't do diffusion on the rotations. They kind of construct the frames as they go using the C alphas agitations. And I think that's another valid approach to it. So what I guess what you're saying is kind of different though. You're saying to still have the frames, but only predict them at the end. And I guess let me think. So would you initialize it at random in the beginning then? The frame, the rotations?
Yes, I guess.
Yeah, I guess that might lead to some weird biasing of the distributions that you learned, because your model will be super dependent on the frames it learns at the very beginning. I'm not sure if, yeah, I guess I don't have intuition what that would be like. Sorry. Okay, someone asked, is there a way to ensure that the protein generated will be stable? So stable in what sense? Do you mean such as having a lot of secondary structure, or stable in the experiments that you run on it? Like thermodynamically stable. Yeah, so I think this gets into oops, shit. Actually, that's fine. So people have been trying to do something like classifier guidance. So you can use a classifier that can predict the stability of a protein and use that to guide the generation of the structure or the sequence. Currently in Framediff and RF diffusion, there's no way to guarantee that's going to be stable, but there are ways to improve its stability by using these guidance terms to help it to be more stable. So yeah, I think it's an interesting research direction to maybe train a classifier that can predict the stability of a protein and use that to guide the generation to be more stable in the experiment conditions.
Thanks.
I have another question that in your report you said that you will work on complex generation. Have you any progress on that you can share with us?
Sorry, say that again. On what generation?
Complex generation to multi-chain.
Context generation.
Complex multi-chain.
Oh, complex generation. Oh,
yeah, yes.
Yeah. So I do have a project that's working on that right now. But since it's recorded, I don't want to say too much. Sorry. Yeah. You can email me and then happy to discuss anything specific you want though.
I just now have
Okay.
one more questions. So when you do inference, do you need to when do inference try to denoise? Do you need to go from the capital T into the T equal to 0, or you just start from the capital T from a small number instead of start from the number you're using in the training steps?
Sorry, what do you mean by that?
Yeah, when you do inference, so you try to denoise from the capital T to from capital T to the T equal to 0. So how large do you set for the capital T? So do you use the same number as it is in the training steps, like a 1,000 step or 2,000 steps?
Yeah, so we do the continuous time variant of diffusion. So like you set the capital T to be 1, so that it's not like discrete time diffusion where you have fixed discretization, but this is a continuous time diffusion. So yeah, you just choose how many steps you want, and that just discretizes time 1 to 0. So that's the benefit of continuous time, that you can discretize it to however long you want the diffusion to be.
Okay, thank you.
Now, I actually have a follow-up question. So, what's the next step? Since we have the IF diffusion, which is really powerful to generate real-world proteins, and we have Framediff. So, what would be the next step? Like complex generation or something else?
Yeah. So yeah, complex generation is something that it's like, yeah, kind of one of the projects that I'm working on. And then I think people have been trying to work on all atom diffusion. So there's like some papers out there that are trying to do both sidechain and sequence diffusion at the same time. So I think that's a very exciting next direction of trying to solve that problem. And in this paper, actually, we tried to do sequence generation at the same time as the structure generation. There might be like some Easter eggs of that in my code base. But yeah, we never got to work. So I'm excited to see what people could think of to do sequence generation at the same time. My collaborator, Brian Tripp, he's been working on motif scaffolding. So he has a new paper out that does like twisted sequential Monte Carlo in doing motif scaffolding. So that's another important direction to improve these methods on.
Yeah. What do you think of the application of diffusion model on proteins? Other applications,
Sorry, on what?
The application of diffusion model on proteins. Yeah, we have the monomer generation, right? And we would maybe have complex generation. But for example, we can apply diffusion model on protein structure prediction. Like, do you think of any other direction applications of diffusion model?
Other applications. Yeah, let's see. So I think it listed out what people have mainly done. So there's like for design problems, for generation, for protein folding. So I think in protein folding case, people are trying to get different conformations to try to do almost molecular dynamic stuff with diffusion models. I mean, I think there's a ton to explore in this application, where how do you do guidance with diffusion models to do different conditional generation? Because there's so many different classifiers that you can use to guide diffusion models that it hasn't been explored at all with proteins, because in protein, there's a lot of different properties of proteins that you want. So someone already mentioned like stability, but binders, how do you like do that with a high success rate and stuff? Enzymes, for instance, we haven't solved how to do diffusion for enzyme generation. And also, there's also modalities as well. So RNA, DNA, people, I think are just starting to do diffusion models for. Cryo-EM, people are trying to do post-estimation for cryo-EM diffusion models. Ligand generation, but people haven't been able to do like ligand and protein generation at the same time. So yeah, it's a very general framework that I think people can try to combine a lot of different data modalities together. It allows for that.
Yeah, thank you.
Jason, I have a thing I want to share with you is that I tested your repo and it's before your final update with the rotation bug fix. And I see that if your scaffold is designed by C alpha MPN, it has better performance than the full backbone atom MPN. Have you see that, or what's your opinion about?
Yeah, I saw it too. At first I thought that was a bug, but it turns out to not be. Yeah, I think there's ways to improve protein PNN basically. Protein PNN, if you read the paper, it's trained with like a very tiny amount of Gaussian noise. It's not trained with a noise that's in SC3 diffusion. So I think because if there's a little bit of error in the rotations with Framediff, then protein PNN isn't going to know how to handle those rotation errors. So it's better in the C alpha when it doesn't know anything about the rotations. Of course, if the rotations are perfect, then protein PNN is going to be happy and is going to do better with the full backbone as an input. I think with the latest update that protein PNN does better with the all backbone atoms. But I also think there's ways to improve protein PNN to make it more robust to these kind of generative models. So yeah, I mean another thing is with generative models, these models are not perfect, right? Like the protein structures they spit out, there's no guarantee that they're going to be like physically perfect. As in, there's still going to be some errors in the bonds. There's still going to be some errors in the angles between frames. So that sort of stuff, protein PNN is going to be a little out of distribution for. So there's a lot of ways to improve protein PNN to be give better, higher success rates for these de novo designs that come out of deep learning.
Okay, thanks. And I want to I also test that with MPN design, but follow up by fast relax and also another MPN design and other fast relax. It improves the Framediff a lot better, maybe 3 times higher success rate, like that.
Oh, cool.
It's
makes
a
sense.
protocol from the late. Also they use it in the alpha diffusion binder design pipeline. Have you checked that?
No, that's actually an idea I had, to do fast relax after the generation.
Okay.
Because I think you and I had the same intuition in that there's like these little errors that's at the end of Framediff that you want to fix.
Yeah,
so
yes.
fast relax would fix that.
Yes, I guess you can try that. That gave a huge better performance.
Nice, that's good to know. Yeah, you should publish it.
Yeah, I guess we have no further questions, right? And if so, we can like end this talk. And yeah, does anybody have any other questions? Well, I guess
feel free to email me or just post a GitHub issue. I try to be responsive to it. So yeah, thanks so much for the invite.
Yeah, thanks so much for joining us.
No problem. Okay, thank you. Yeah.